Hello everyone, in this lesson, we will study the pathogens of plant infectious diseases. The pathogens of plant infectious diseases include fungi, phytopathogenic prokaryotes, plant viruses, plant pathogenic nematodes, and parasitic seed plants. 5 main kinds. First, we will study the pathogen fungi. A fungus is an eukaryotic, package-bearing, chloroplast free eukaryote, including mold, yeast, mushroom, and other fungi known to humans. There are more than 380 kinds of tea plant diseases in the world. Medicine caused by fungi accounts for more than 80% in daily life. We have the following common fungi. First, aflatoxin produced by peanut mildew belongs to fungi, which may cause cancer. Second, medical antibiotics such as penicillin, flavomycin, cephalosporin, and so on are drugs produced by fungi. Third, some valuable medicinal materials are fungi themselves, such as Poriococcus, and so on. The fourth, edible mushrooms are fungi, such as Lentinus adus and Hericium arenosus, ambusan, fungus, tremula, and so on. Take a look at the vegetative body of the fungus and its metamorphosis. Then, what is the vegetative body of the fungus? During the vegetative growth stage, the fungus is used to absorb nutrients and growing institutions, called nutritative bodies. The vegetative bodies of fungal types are small filaments, which are called hyphae, and the aggregates of hyphae are also called mycelium, in which the hyphae are mostly colorless and transparent. The hyphae are divided into 2 parts, namely the septate hyphae of higher fungi and the septum-free hyphae of lower fungi. The mycelium radiates from one point to all sides and usually forms round colonies in the culture medium. What is the nutritional metamorphosis? The mycelium of fungi is generally dispersed, but some fungal hyphae can gather together under certain conditions to form bacterial tissue. There are 2 types of fungal tissue: filamentous tissue and pseudo-parenchyma tissue. These 2 tissues can form abnormal vegetative structure, many sclerotia and fungal cords, and so on. Sclerotia is a kind of hard dormant body formed by 2 kinds of fungal tissues. The sporangium is a cushion formed by 2 kinds of bacterial tissues, or by mycelium, and part of parasitic tissue. The fungal cord is a rope-like substance composed of parallel mycelia and the vegetative form of fungi. The vegetative form of fungi includes asexual reproduction and its spore type, sexual reproduction and its spore type, and quasi-sexual reproduction. First, let's take a look at asexual reproduction and its spore type. The basic characteristic of fungal asexual reproduction is vegetative reproduction, which is produced directly from vegetative bodies, asexual spores. They produce various types of asexual spores through 4 ways: cleavage, budding, fragmentation of budding, and protoplasm. Then, let's take a look at asexual reproduction and its spore types. Sexual reproduction of fungi is a way of producing offspring through nuclear binding and meiosis. Spores produced by sexual reproduction are called sexual spores. There are the following kinds of sexual spores. Finally, let's take a look at quasi-sexual reproduction. In addition to asexual reproduction and sexual reproduction, a few fungi also carry out another way of reproduction. That is, quasi-sexual reproduction, the life history of fungi. The process in which a fungus begins with a kind of spore, goes through the stage of growth and development, and finally produces the same kind of spore, which is called the life history of the fungus. The classification of the fungus. The Ainsworth classification divides the fungus into 5 subphyla. The nomenclature of fungi. The fungal nomenclature adopts the double nomenclature. The former name is the genus name, and the other is the species name. The first letter of the genus name should be capitalized, and the species name is all lowercase. For the official scientific name of fungi, it is necessary to add the name of the person after the genus name and the species name. If the naming person is 2 people, the surnames of the 2 people should be connected by the conjunction and so on or and. The names of the first namer should be written in parentheses. For example, the pathogens of Colontotulum camilli. Some pathogenic fungi are named according to the morphology of spores in a sexual stage. Before a sexual stage is found, next we learn about plant pathogenic prokaryotes. First, look at the general morphology of plant pathogenic prokaryotes. The morphology of bacteria is divided into three types, namely globular, rod-shaped, and spiral-shaped. The growth of bacterial marginal hairs in three ways, namely single, polar, and peripheral flagella. Bacteria are mostly colorless and transparent. Their morphology and structure can only be better observed after dying. The most important thing for bacterial staining is gram staining, which is stained with crystal violet, treated with iodine solution, and then washed with alcohol. Those who do not fade after washing are gram-positive reaction. We use G+ to mark it. Those who do fade after washing are grander-negative reaction. We use G- to mark it. Next, look at the classification of plant pathogenic prokaryotes. So far, there are 28 main genera of plant pathogenic prokaryotes, which are divided into 6 main genera, as shown in the picture. Due to time constraints, in this class we can only learn about pathogenic fungi, plant pathogenic prokaryotes, the remaining contents, plant virus plants, pathogenic nematodes and parasitic seed plants, 3 aspects. We will learn in next lesson. That's all. Bye.